Okay, so let us start our class today. So in the last class, we were going through the fermentation of the glycerol. So glycerol, as we know, so let us briefly look into what we have studied in the last class, and then we will be discussing today the fermentation process, the steps, how we carry out, the media composition, and what are the downstream processings that are being used to purify and obtain the glycerol fermentation. And finally we'll look into what are the applications of the glycerol. So glycerol, as we have already discussed, can actually be produced by both the chemical and the biochemical routes. And if you see, for example, the production of the biodiesel, which is the fuel which can be used as a fuel additives, there is a tremendous production of the glycerol as a byproduct. So that is a one step on the chemical route by which the glycerol is produced. But the glycerol can actually also be produced by the fermentation processes using different type of the carbon sources. So earlier, sometimes in the earlier times when the discovery of when the production of the glycerol was taking place, people were using the propylene to convert the propylene into the glycerol and through the chemical routes. However, as we know that the petroleum feedstock and the cost of that of the petroleum feedstock and all those derivatives are increasing day by day. So therefore, there is a shift in the tendency is to use the microbial routes or fermentation routes for the production of the glycerol. So we have seen in the last class that there are different ways by which or strategies by which the microorganisms are modified so that we know the overproduction of the glycerol occurs. So because if you see what is the main objective, for any industrial process is to have the higher yield as well as the higher the purity and the quality, so that the number of the downstream processes that is required to purify uh, should be as low as possible. So we would like to sometimes some approaches of like metabolic engineering or genetic engineering is carried out on the microorganisms to improve their the productivity of the glycerol. Okay, and the different bacteria, yeast, and different microorganisms are used, because if you see the bacteria, yeast, and other microorganisms, the rate of their growth is different for the different microorganisms. Therefore, different approaches are being used to carry out the overproduction of the glycerol. Now, so we have seen that what are been what are the some of the recent works, or if you see the technological advancements that has been carried out, especially in the glycerol fermentation, like is the development of the osmotolerant yeast. So osmotolerant yeast means that the yeast has been genetically modified in such a way that it can able to withstand the high osmotic pressure, and that is basically just generated during the production of the glycerol. So glycerol generally have the high density, and it actually, if its concentration increases in the fermentation broth, it is going to create osmotic pressure or osmotic stress on the microorganisms. Okay, so if we have a osmotolerant yeast, it was found that the productivity was as high as 130 gram per liter, and with a yield of 63%. Okay, so there was a significant improvement in in the production of the glycerol. And however, other strategies of of improvement in the production of the glycerol was using a novel downstream processing. So this is called the carrier distillation technique, by which it was observed that almost 90% glycerol was obtained once that the glycerol, general glycerol that we obtain is dissolved in the fermentation broth. So we have to have a a very good the downstream processing techniques to recover them, and the carrier distillation technique is one of the approach by which we increase the efficiency to as high as 90%. So you can see there are different ways, either by metabolic engineering or by genetic engineering we can improve the yield. Also there are some other chemical engineering approaches like the downstream processing. We can modify them to improve the yield and the extraction of the glycerol. So so those are the different ways how we can improve the production of the glycerol, and there are other and if you look into the biosynthesis pathway, so we have looked into the biosynthesis pathway earlier, like if we start from the glucose, so glucose to the glucose 6 phosphate to the fructose 6 phosphate. This is a very basic the part of a section of a glycolysis pathway, where the glucose is converted to 6 phosphate and to the fructose 1,6 biphosphate. And after this there is the branch point that we can call that. After this it mitigates to into, or it it breaks into two steps of production of a dihydroxyacetone phosphate or the glyceraldehyde 3-phosphates. Okay, so and this step where the glyceraldehyde 3-phosphate can actually be converted into the dihydroxyacetone phosphate. Okay, and so if you are targeting for the production of the glycerol, we have to have the higher concentration of this particular metabolites, that is the dihydroxyacetone phosphate. So we make sure that the enzymes that are required for the conversion of the fructose 6 phosphates are actually upregulated, as well as if there is a production of the glyceraldehyde 3-phosphate, then we make sure the enzymes are present which can irreversibly convert the glyceraldehyde 3-phosphate into the dihydroxyacid phosphate. So we will look into that, like how what are the strategies by which actually we can improve the production of the precursors, especially the dihydroxyacetone phosphate, so our main target will be to improve the concentration of this precursor. So this is the main objective. And once the precursor is at a very high concentration, then automatically this DHP, dihydroxyacetone phosphate, will be converted back to the glycerol in the presence of these two enzymes. So we will look into that. So generally, if you see the production of the glycerol, it has been carried out through the three basic processes. So depending upon, first of all, if depending upon the microbial or microbes that are used, if yeast are used, so generally there are three process. First is called the sulfite process, second the alkali process, and third the osmotolerant yeast process. Okay, so each of these process have their characteristics, and by using another microorganisms called like the algae, so which is called as the algal process. So we will see what are the characteristics for each of them, and what are the advantages and the disadvantages. So from that understanding, we can actually then design your fermentation process, because each and every process have their own particular characteristics and the properties. So if we look into for example means the first method. So the first method generally, as you can see in this particular biosynthesis pathway, here you can see that these two steps, that is the production of the glycerol and the production of this ethanol, they are competing with each other. And if your target product is the glycerol, then we have to make sure that this particular the pathway should not be should be inhibited. Okay, and then only what happens, whatever the glyceraldehyde is there, it will convert it to the DHP, that is dihydroxyl phosphate, and we will have the more conversion of the glucose to the glycerol. So to carry out those things, there are several strategies how we can we convert this glyceraldehyde 3-phosphate to the DHP production. So one of the strategies is by the trapping of the acetaldehyde by the bisulfide ions. So bisulfide ions, actually what it does, it reacts with the the hexose, so and then it produces itself the acetaldehyde bisulfite with the release of carbon dioxide, water, and glycerol. So basically, um, the acetaldehyde that is there is actually it's serve it's it helps into the accumulation of the NADH. Okay, and that NADH actually helps in the reduction of this DHP phosphate to the glycerol 3 phosphate. So basically, what it does is the if you see the bisulfite ion, so bisulfite ion and the acetaldehyde moiety, actually that is acetaldehyde moiety means the acetaldehyde that is present in this particular structure of the glyceraldehyde 3 phosphate. Okay, so these bisulfite ions actually react with the or it bonds with this glyceraldehyde molecule, and it helps in the conversion of this G3 glyceraldehyde 3 phosphate into the dihydroxy acetone phosphate. Okay, and that is carried out in the generally it is carried out as in the cytosolic NADH, and it helps. How does it carry out? How means how it is being catalyzed? It is catalyzed by the accumulated NADH that is present. Okay, in the fermentation. So this is how actually the conversion. So this presence of the bisulfate ions that is there during the fermentation process helps in the conversion of this glyceraldehyde 3 phosphate to the dihydroxy phosphate. Okay, so this is the one of the approach through which it is done. Then there is second approach of using, similarly, there is second approach of using the alkali processes. So, so generally, if bisulfite is used, it is called as a sulfite process. And if, for example, for the similar conversion, the alkalis are used. Okay, so alkalis are basically used in during the anaerobic fermentation process, and so in this case it is called as a alkali, it is called as a alkali process. So another approach of making sure that we have the higher concentration of the DHP, that is hydroxyacid phosphate, and we convert this glycerol phosphate to this DHP. It is by maintaining the fermentation conditions. So one of them is to maintain a fermentation condition of almost the pH values above 8, and it has been found that the optimal concentration for the increased activity of this aldehyde dehydrogenase, which is the enzyme that is responsible for the conversion into dihydroxyacid phosphate. So it should have an optimal pH has been found out to be the around pH 8.75. Okay, so because of this pH condition, it actually generates an oxidation environment, and it generates a molecule of the NADH, which is required for the oxidation of this DHP, that is of this the glycerol 3 phosphate into the glycerol 3 phosphate. Hello, am I audible to everyone?
Yes, sir.
Okay. I think there is some network issue at ITBHU, possibly because of some maintenance work. So let us quickly, go through the slides. So is it visible? Is the slide visible?
Yes, sir.
Yes.
Okay. So if there is any problem ahead, please let me know. Okay. Okay. So okay. So we were discussing on how we you can actually tune or tune this fermentation process in such a way that whatever the intermediate that is being produced can be so it is basically nothing but any enzymatic reactions they are reversible. So how so main thing is that how we can bring the reactions towards the production of this DHP, that is dihydroxynosine phosphate. And for that bisulfite ion, as we discussed, the bisulfite ion is one of the ways how we can convert this glycerol 3-phosphate into the DHP. Another way is to use the alkali based method, the alkali process. So generally, this type of chemical additives, which is used to convert for the backward and the forward reactions, so those are actually called as a steering agent, because these steering agents, so like we have talked, the steering agent like sulfide. So steering agents are required, which can, for example, tune the pathways. So either if you would like to have a forward chemical reaction or a backward, so the steering agents are added so that your desired the biochemical intermediate products that are there, that can be produced. So if we so this is a very basic understanding that everyone should know, that on the basis of the steering agent like sulfite or the alkali or these are the two most of the used steering agents that are there. So this is called as a sulfite process. So if we compare the three processes, if we look into that, so microorganisms are generally that are used is our yeast. So we have mostly Saccharomyces cerevisiae that is being used for the production of the glycerol. And in this case of the osmotolerant yeast, we have the genetically engineered yeast, in which it has been engineered in such a way that it is able to withstand the high osmotic pressures. And osmotic pressure against what? Against the high concentration of the glucose or the sucrose that is being going to be used during the fermentation, as well as against the glycerol that is being produced with the fermentation time. So if you see the steering agent, yes, the steering agent is required for the sulfide process, which is not required for the osmotolerant process as well as the algal process. And if you look into the algal process, the organisms are halo tolerant, algal, CO2 and light. So as we have know that algae are they use we have seen in our first classes that what is the difference between the bacteria, viruses, algae, yeast, and so on. So in that, I think if you remember, if you recollect, you can. So the substrate that is required for the growth of the algae are the CO2 and the light. So because they use the photosynthesis pathway for their biomass production and as well as other biochemical production. If you see the oxygen requirement, so generally it is the osmotolerant and the algal process are aerobic. That means it fermentation is carried out in presence of the oxygen. However, in the case of the alkali process or the sulfide process, it should be carried out in the absence of the oxygen, and it is the anaerobic process. So, and if you see time, fermentation time, it is 4 to 5 days for the sulfide process, 5 to 7 days. And generally for the algae, the growth rate of the algae are generally slower. So generally it takes 10 to 14 days. But if we genetically engineer these microorganisms, the time frame is the least. So this is a very big advantage when we have a genetically engineered or modified the microorganisms. So we can ferment, we can the fermentation time is as low as 3 to 5 days. So if you see the fermentation control for almost all the processes, it is difficult, only except the genetically engineered ones. And the formation of, generally during the fermentation process, one of the thing that is very much concerning is how, the contamination is there. So as I have already discussed, that when you are doing the fermentation, we make sure that it should be, carried out in, it should be in aseptic conditions. So that's why we, what we do is we sterilize your growth media components, and every time we maintain a sterilized condition. So that is very important for whenever you are carrying out any biochemical process, that you have to maintain a very clean environment. And generally, though means you maintain the clean environment, but as you work, there is a high possibility or chances of getting contaminated from, getting the contamination inside your your fermenters. And suppose you are, if you are working with a very large pilot plant scale fermenters, then what happens is that, so if the microorganism is prone to the contaminations, then if there's a contamination, then the whole batch has to be discarded, and that leads to a huge, the financial losses. So so when we genetically engineered or modified this type of stain, we make sure that, the contamination problems or the infection problems are as seldom as low as possible, and that can be obtained in, the algal process and the, yeast based processes, especially if you have the osmotolerant yeast. If you see the conversion efficiency, you can see that if through a traditional process of like the sulfide and alkali, it is 25% conversion of the sugars that we use into the glycerol. And but if you go for the osmotolerant one, it is very high. So we can see that 60% of the glucose source or the sugars that is being there, that is converted, and it is having a very high concentration of, the glycerol, that is 110 to 130 grams per liter. And if we compare it with the traditional process, which is 30 to 40 grams, so you can see that almost there is an increase in, the three times production of or productivity using these genetically engineered organisms. And yeah, if you see the algae, in that case, it is significantly very low. Okay, so it's a very low production, as well as the time of, the production of the glycerol by the algae are very high. And because of that, because of these two disadvantages, it is generally not used in the industrial scale fermentations. Also, after the fermentation process, one of the thing that after the fermentation process, after you have, extracted the glycerol and purified it, then the main thing is that whatever is left behind is a byproduct, the cell biomass, the growth media component, so on. So it is very much important, like because when you are working in the industries, we are working in tons and kgs of volumes of of the components or the media components. So we have to develop a strategy in by which we can, uh, dispose of your, of your these byproducts. So the environment pollutions are generally very heavy for these traditional processes compared to the genetically engineered one. And like we already discussed the commercialization, so sulfide process have been, widely commercialized. Okay, and but the challenge is, it's still uneconomical, and alkali process have not been yet, and as well as algal process, because of, the advantages of high fermentation time and low productivity. However, the genetically engineered osmotolerant yeast processes have been, commercialized, and they are found to be economical, and because of which, the shift in the, the production of the glycerol production is taking place using the microbial fermentation route, in comparison to the chemical processes. Because it is found to be economical and, and cheap, and you can use not only, the glucose as a substrate, and wide different types of the substrates, maybe, the agricultural waste and those derivatives can be used for the production of the glycerol. So this is overall actually tells you about a brief summary on, different processes that are there for the, the glycerol production. And if we see the microbial stains, that are the microorganisms that are being used for the glycerol production, so each and every microorganisms have, a different production capacity, and there are different processes, the scale at which it is being, produced. So if you see this particular table, it compares with the different microorganisms like the yeast, bacteria, and the algae. Okay, and it tells you about what is the substrates that has been used, and the process for the scale at which that has been produced in a fermented reactor, and what is the concentration of the glycerol that's been produced, and how much the sugar has been converted to the glycerol, and what is the productivity. And whenever we talk of the productivity, we have the gram per liter per day, that day how much of gram of liter, how much of your glycerol gram per liter is produced. So if we just see and compare the different, the yeast, for example, like we have the Saccharomyces cerevisiae, then we have Candida magnolia, P. farinaceae, and Glycerinus. So here we can see that if we use the readily metabolizable sugars, so glucose we know is can actually be easily metabolized compared to the other mixed sugars, like in the case of the molasses. So we can see that the using the the glycerol glucose as the as a pure carbon source, there is a significant improvement, a significant difference in the productivity. So using a pure glucose always gives the high production or high concentration of the glycerol in the fermentation broth, in comparison to the molasses, because molasses contain different the oligomers of the glucose. It may be sucrose or other derivatives are there. So the the conversion rate by the microorganisms is significantly lower when we have the mixed sugars. If you see the compare the different the processes, so generally here it combines the batch sulfide process. So we have either batch in the sulfide process, we have either batch type or a continuous type, depending upon the reactor system that you're using. If you have a batch fermenter, it's a batch sulfide process, or if you have a perfusion reactors, then we have the continuous sulfide, or we have the fed batch process. So if you can see, if you compare these three, the batch, continuous, and the fed batch, so generally the fed batch is having the highest concentration of the production. And why is it so? It is because of the glycerol that is being produced. Okay, that is continuously removed, because as I told you, this glycerol exerts osmotic pressure, and that it exerts osmotic pressure on the membrane of the cell. And if its concentration is very high, what will happen, the rupture of this outside membrane will actually takes place, and it will lead to the killing of the cell or the cell death. So if whatever the glycerol is being produced, if it can be taken out with time, and to make sure that the concentration doesn't goes above a threshold concentration, so by making that thing in a fermentation, in a fermenter, we can actually improve the productivity of your glycerol compared to the other processes with the batch process. Okay, so that is why the fed batch is widely used, and is one of the it is advantageous compared to the batch and the other processes. If you see the other, so this is the overall, this table summarizes actually that uh, the overall improvement in the production of the the glycerol concentration. And if you see the conversion of how much of the sugar is being consumed and how much of it is being produced into the glycerol, we can see it is almost similar, except in the case of when you have the glucose, where we have higher conversion of the glucose to the glycerol, and we have obviously the higher yield. And another thing to note about is the what is the scale at which it is being produced. Okay, so you can see that till date, the maximum glycerol production has been carried out in the reactors of volume of 50,000 liters. Okay, it is starts at the from the scale, we have the sec flux, and then we move up to the pilot scale into like 12 liters, 2.5 liters, and then this is the larger scales where we can produce it at a 50,000 liters. So we generally this step of the moving from the lab scale in the sex flux to the pilot scale process, and then to the industrial scale process where we produce. So this glycerol production has actually been already commercialized at a commercial scale, and we can see that using this particular, if glucose is used as the pure source of the carbon, we can have a very high conversion of the glycerol. Along with that, we have the very high conversion of the sugars into the glycerol with a very high productivity. Okay, so this gives brief overview on the different microbial stains, the different substrates, different process scale, and their effect on the production of the glycerol, conversion of the sugars in the glycerol, and their productivity. Now let us look into the growth curve of which is very much important for any fermentation process. For so because until now I think we have always seen and come across this growth curve, which actually gives you the information regarding how the product is produced with respect to the time. Then how is the we also see how is the biomass is produced with respect to the time. And another information that we see is how the substrate being, for example, the it may be the molasses, glucose, or whatever the substrate it is there, how the substrate is being utilized with time. So these three parameters, these are informations that we obtain from the growth curve, be it for any biochemical production. And this evaluation of this with respect to the fermentation time is very much important when we would like to design your fermenter, or how we monitor the fermentations, how we carry out, and how it has to be monitored or optimized in such a way that we always obtain the high concentration of the product that with respect to the time. So if we see this particular plot, so you can see this, the box, the hollow boxes actually shows how the glucose concentration is changing with time. So it starts with around 22 gram per liters of the glucose, and we can see that the glucose concentration is rapidly decreasing with the time, and it almost goes zero at almost those 72 to 80 hours. And if we look into our pathway for the glycerol, which is this diamond, so if we look into that, we can find that it is increasing with the time, and until the 48 hours, okay, it is almost it's stabilizing, and we cannot see a significant change uh, in the production of the glycerol. However, along with the glycerol, there are other derivatives being produced. Like we have already seen that there is a bypass of ethanol production. Along with that, acetate and lactate are also being produced. So these are, so as I told you, these are the major byproducts that are there. And we have to make sure that these byproducts are actually utilized by the cells in the cells towards the production of the glucose. Okay, so if you see the byproduct formation, especially the lactate, so lactate formation takes place until the 40 hours. However, after that, the concentration is decreasing. That means that the lactate has been utilized for the production of the glycerol. And similarly goes with the acetate. So here acetate is, you can see the acetate is being produced, but it stays within the media. So this acetate is actually not being used for the production of the uh, for the production of the glycerol. Okay, so whatever the and again it's the ethanol, you can see that it goes and maximum until the 32 hours, and then its concentration is decreasing with time in a stepwise manner. That means the whatever the ethanol has been there, it is being used for the production of the glycerol. So both these derivative, the ethanol and the lactate, are actually used up for the production of the glycerol. And so that is how you can see that if you see the utilization or the production of the glycerol graph, uh, this for example this Laban bar, it is rapidly increasing until this point, and there is a slight increase, and then it stabilizes. So first what is so if you see you can convert, you can break this graph into 2 points. Until 48 hours, what is happening, whatever the glucose is being the raw sugar is there, that glucose is converted into your glycerol. And after that, whatever you can see, there is a bit increase in the production that is coming from the the byproducts that are there. And byproducts are what, the ethanol and the lactate. Okay, and during this step we can see that there is a significant decrease in the concentration of the ethanol, as well as the lactate is occurring. So what is happening, because two component, the ethanol and lactate, these are going to be converted to the glycerol, and we can see actually increase in the production of the uh, of the glycerol. So generally, it can be classified into the two-step production. One is directly conversion of the glucose to whatever it is being present in the fermentation media to the glycerol, and then conversion of the byproducts that are being there, the ethanol and the lactate, into the glycerol. Okay, so this actually how the growth of or the production of production of the glycerol takes place with the fermentation time. Next, let us look into the overall process flow diagram. Okay, so this actually shows you the process flow diagram on utilizing the carbon sources for the production of the glycerol as a final product. So here we have we can classify it into the different steps. The first step that is there is utilizing the agricultural sources. So for example, we have a corn starch. So this corn starch, from this corn starch we have to extract, from the starch we have to extract the sugar derivatives. So what are the sugar derivatives which can be metabolized by the microorganisms? So they are nothing but the glucose. So starch, we know, it's a long chain of the glucose molecule. So we have to break those long chains into the smaller units of the glucose, and that is what is called the liquefaction or saccharification. Saccharification is a process by which we break down this starch molecule or the long polymer molecule of this polysaccharides into the monosaccharides. So when we have, so this is a glucose liquor because it is having the pure glucose, along with that it is having the mixed sugars. Okay, so it may contain the oligomers of the glucose molecule and so on. So glucose, we know, it's a six carbon ring like structure, right? So it can be, so when we call the glucose liquor, it can be a monomer, or it can be a oligosaccharide. That means two glucose molecule can be attached with each other, and that may be produced during the fermentation. So this is how we first step start with the enzymatic conversion of this cornstarch to the glucose. And generally we use different enzymes. So we have already seen the enzymes that are being used. If you are having the cellulose to the glucose, if you want to convert that, we have a cellulase enzyme. So if you have a starch, we have a different enzyme. Similarly, if you have a lactose, you have different enzymes, and so on. So everyone, so depending upon what is the your initial substrate, you can select a particular enzyme for the conversion of that polysaccharides to the glucose derivatives. Now next step comes in the development of the inoculum. So in the inoculum, what we are doing is we select the the microbial strain that is there, that is being preserved, and so we use that strain to culture that strain into the high biomass concentration. So we use the strains that are being preserved in the different slants that we have already studied in our earlier classes. So we take out the preserved microbial cultures, and we grow them in a particular inoculum media, and we make sure that we obtain a particular concentration of the biomass that can actually be used as a seed culture in the fermenter. Okay, so we have already seen for all our biochemical processes that we make sure that we get an optimum concentration of the biomass, and then it is being added into the fermentation media, where we have fermenters in which the growth media or the production media components are there. So once in the fermentation we have the different production media components, then we the sterilize that, and we inoculate whatever the biomass that we have produced in this particular inoculum step. We inoculate them and we start the fermentation. And in this case, mostly the fermentation, if you see the glycerol production, uh, depending upon which process we use. Okay, so it can be anaerobic. If we are going with the sulfite as well as alkali process. If we have a osmotolerant yeast process, it can be aerobic, or if it is algal, it will be aerobic. So depending upon that, we supply the sterilized air. Okay, and the concentration of it is varied. And after, during this process, what is happening, the production of glycerol takes place. Okay, and once the production of glycerol is taking place, so generally glycerol is mixed with the fermentation broth that is there after the fermentation. So we take this particular broth and we carry out the the different downstream processing. So downstream processing, first process is the filtration. The filtration is used to remove the cell biomass. So what is happening, like in the fermentation system, generally after the inoculation with the microbial, with the yeast, what is happening in the fermentation, the microbial biomass or the cell concentration will increase, right? And it will be in some sort of a solids that will be dispersed within the fermentation broth. So we have to remove that cells by by the very simple filtration, and we obtain like a very yeast biomass, and they have their different utility as a animal feed source and so on. Okay, after that the next step is the concentration. So we have to concentrate the solution, because the growth media components have the mostly have the aqueous environment in which the media components are dissolved in the water. So what we do is we evaporate, carry out the evaporation and the vacuum. So we have seen this evaporation to concentrate the solutions. Okay, and after that we carry out the vacuum carrier distillation, which is nothing but to distill out the glycerol. And once the glycerol has been distilled out, then what we do is we actually carry out the decolorization. So generally the glycerol, if you see the in the labs, if you see a glycerol solution, the glycerol is a transparent in nature, but because of the presence of different growth media components and the biomass and other components, they have a slight yellowish color after this vacuum carrier distillation. And to decolorize those substances, we use decoloration by the activated carbon, followed by the filtrations to remove these activated carbon particles. Okay, and then purifications by the resins, and the final evaporation to get the concentrated solution, which we and finally what we obtain is the pure glycerol. And generally we have 99.9% of pure glycerol, and rest we have other impurities like water and so on. So this is how we actually, gives you a overall summarized overall view of how what are the different downstream processings, and why each and what is the importance of each and every downstream processing steps that are required, okay, for the glycerol production. And along with that, what is the the fermentation conditions, how to make the inoculum. And if so, here we are starting the glycerol production using the cornstarch. So as I already told you, the utilization of the biomass or the waste biomass can actually help in both utilization conversion of the waste into the value added products, as well as it will reduces the overall cost of the fermentation. So this is why biomass has to be used, and then requires some additional process. Okay, so like we have here the enzymatic treatment. It may be a chemical based treatment. So we have already seen those things in our last earlier classes. So this is how the overall process flow diagram is there for the production of the glycerol using the fermentation approach. And another thing, that so now let us we look into what are the, so this is the fermentation conditions, but what are the the what optimized conditions, and what are the factors that are taken into consideration when we carry out the glycerol fermentation. So there are some of the environmental factors, so which are there, and they are the temperature. So these are the factors which are being the optimized, or which are being monitored during the glycerol fermentation. So one is the temperature of the fermentation system, aeration, which means how much the concentration of air is being dissolved. What is the concentration of the phosphate ions in the growth media, nitrogen concentration, that is some sort of ammonia or nitrate, and the sugar concentration. So as I already discussed, the sugar concentrations, that is the glycerol, that is the glucose, or any other initial sugar concentration, how much this there, and what is the osmotic pressure on the cells. So these are the because of the production of the glycerol. So these are the the parameters which actually monitor the rate and the yield of the glycerol production. So if you would like to optimize and have a maximum production of the glycerol, you have to optimize these parameters. Okay, for the case of the temperature, which has been found like it varies with the different microorganisms we use. However, it has been observed like if we are using the candida glycogenesis, the optimum temperature is lies between 29 to 33 degrees Celsius. And if you have the Gymnovius hydroxy being the or the cerevisia or candida magnolia, they lie in between the optimal temperature is in between the 30 degrees and the 35 degrees. So here you can see that almost it's near about. So in a temperature range of almost 30, in between 30 to 35 degrees is suitable enough for the production of the glycerol, and a higher temperature, suppose at 40 degrees Celsius, or for example, they actually means the production of the glycerol is inhibited. And another thing is, the degree of aeration. So also the degree of aeration, what is the parameter to evaluate that is the how much of the dissolved oxygen is there. Okay, so that requires optimization, as well as how much the concentration of the phosphates are there, and that needs to be optimized whenever you are carrying out your glycerol fermentation process. The other thing is that optimization, not only in the fermentation process is important, but optimization in the extraction of that is very important. Like we have talked about that we have a carrier distillation process, which has shown to which has led to improvement in the production by almost 99%. Okay, so so depending upon that, so there are like different processes. One is using of the ion exclusion and the ion exchange chromatography, and it has been found that is able to improve means it able to remove the ionic impurities that are present, which may ionic impurities means like suppose in a fermentation media you have used some salts like ammonium nitrate. So ammonium nitrate, what will happen in the growth media, it will dissociate into ammonia and the nitrate ions, ammonia being positive, nitrate being a negative ions. So we have to remove these ionic impurities. So ion exclusion and ion exchange chromatography are one of the very well known approaches by which you can recover these ionic impurities by by based on the charge based interactions. Okay, so we will not go into details of what is ion exchange chromatography. It is just a charge based interaction where positive ions move towards the negative one and gets accumulated and separated out, and similarly negative ions will accumulate on the positive surface. So this is how we generally remove those type of ions. And using these type of approaches, especially this ion exclusion, ion exchange chromatography, or the the carrier distillations techniques, it has been found that it will help in improvement in extraction. Because if you see the traditional vacuum distillation, only it was observed that only 50% of the glycerol that is there can actually be recovered. Okay, so to obtain the 99% of recovery, we have to go for the carrier distillation techniques. So these are some of the the advancements, especially in these downstream processes that has been carried out for the glycerol production. But if you see the means after the glycerol has been produced, then what is the application of that glycerol produced from different agricultural waste byproducts, or for example in the chemical based approaches during the biodiesel production. After that, what is the the glycerol cannot be actually directly added. So if you look the biological applications, it cannot be added directly into the food additives, so it has to be converted into some form. Okay, so what are those forms or what are those applications? So they are like many type of amino acids. Okay, especially the L-tyrosine, and then we have L-serine, other type of the D-lactate or L-lactate. So this lactate or the lactate acid and these type of amino acids, they have several applications in as a food supplement or the feed supplements to the animals, and especially in the human we have a lot of food supplement applications of these derivatives. So generally, the glycerol, which is all a byproduct from the industries, or glycerol that is being produced from by the microbial culture, they are processed into another type of chemical or the biochemical based system, where mostly chemical based system or the biochemical based systems in which we are obtaining the different sort of amino acids or different types of the lactate derivatives that are there. Okay, and they find their potential applications in in the food supplements. Some other application of the glycerol is like we carry out a dehydrogenation of this glycerol. Okay, this we remove means we carry out is for use it as a fuel derivatives and so on. So there are several applications. However, if we look in the area of the biochemical areas, most of them is used for the amino acid production, and this whatever the amino acids being produced, it is used as a supplement, as a food supplement, because we have now several products where we make sure that the the amino acid component, the protein components are present at an adequate quantity. So these type of products are added, or amino acids are added, to make sure that you obtain a proper nutritional value product. So this is the overall that is the application part of the glycerol after it is being produced. However, this actually tells you here now this the overall process. So we have looked into the very basics of the glycerol. What are the advancements that are being made into what is the biosynthesis pathway, and what are the the techniques or processes by which it can be produced, and what are the microorganisms, how what are the different substrates that are there, what are different processes, and how much is the general yield and concentration or productivity of the glycerol during the fermentation process. And then we have looked into the growth curve and how it is changing with respect to the fermentation time, finally coming to the process flow diagram. And here we can see that we can use different biomasses to convert them. Next step being the inoculation, third being the fermentation, and the fourth being the extraction of your of your produced product from the fermentation broth. And we see that in the case of the extraction process, we have to select appropriate methods. And if you would like to optimize the fermentation process, these are the main parameters on the basis of which the optimization of of the glycerol fermentation will be carried out, so that we obtain the maximum yield and the concentration. So this comes to the end of the glycerol fermentation. And from the next class, we will start a new topic on the very interesting topic on single cell protein production and the mushroom. So single cell proteins are most of the, if you see in the space application, the food additives that are used are based on the single cell protein. So let us look into those from the next class. So anyone has any questions or doubts? Anyone? Hello, am I audible? Hello.
Yes, sir.
So if no one has any questions, then thank you everyone. Let us meet in tomorrow's class.
Thank you, sir.